Welcome to the lecture. In this lecture, we are going to discuss the nucleus activity of copper phenylthronin complexes. Now, let's discuss. As we know that the nucleus activity is nothing but nucleus is an enzyme which can rupture the desired portion or the desired part of the DNA strand. So here this copper phenylthronin complex is an artificial nucleus which can cleave or which can rupture the portion of DNA strand. Let's see. The artificial nucleus activity by orthophenylthronin complex by copper orthophenyl complex cleaves DNA by oxidatively degrading ribose from its binding site within the minor groove of DNA. As we know that first the complex will react with DNA strand by the groove, and this groove so here likewise copper orthophenylthronin complex is also react with this DNA by the groove, by interacting with groove and then by strand. Since the coordination complex is small and it detects local structural variability of this feature of DNA helix. So generally this complex is reacting with DNA strand by the groove and then by the strand. So the another characteristics or the activity of this copper phenylthronin complex is that copper orthophenylthronin is active and cleaves DNA by intracellularly. Now a range of various copper phenylthronin complexes, that is including in copper phenylthronin complex type cysteine as potential lead compound, potential lead compound for the therapeutic as potential and biochemical application, therapeutic and biochemical application. So this copper phenylthronin complexes have therapeutic and biochemical applications. Complex is believed to cleave DNA by various bond copper bind oxidant like OH and copper with free OH. Here this OH is act as a free radical in overall process. Free OH possibility to play important role in overall process. Then that this free OH in overall process, that is a interaction of copper phenylthronin complexes with DNA strand generally done by this OH free radical. So these are all about the activity of copper or the phenylthronin complexes. Now next, what are the reactions of metal complexes with DNA? Let's see. Now let's discuss the reactions of metal complexes with DNA. As we discussed earlier, that the first reaction is coordination. More prevalent among with DNA are those involving coordination between metal ion and nucleophilic base pairs. The second reaction is hydrogen bonding. Planar heterocyclic aromatic ligand such as phenylthronin, pyridine can stack in between the base pair and stabilize through dipole-dipole interaction. Now, fundamental reaction with nucleic acid. We can say that those involving redox reaction involving metal ion which mediate the oxidation of nucleic acid and the second, this is the A because it's categorized into two parts, and in B those involving coordination of metal center sugar phosphate. So these are all about the reactions of metal complexes with these are all about the reactions of metal complexes with DNA. Thank you.